Welcome to the next class of biology. Here, the first question is the enzyme recombinase, which is required at which stage of meiosis 1? The most important chapter of first year, that is cell cycle and cell division. Cell cycle and cell division. In this chapter, why this is most important? It is because it is related to the all other chapter, such as cells, the unit of life, then biomolecules, the most important chapters such as genetics, genetics 1 and genetics 2. So, cell cycle and cell division. In this chapter, the two type of cell division process, directly the meiosis and mitosis. The two cell division, mitosis and meiosis. You know the difference between the mitosis and meiosis. Mitosis is an equational division. Equational division. And here the meiosis is the reduction division or reduction division. Equational division, mitosis, meiosis is a reduction division. Why mitosis is known as equational division? Because the product, the end product of mitosis, they are two daughter cells. And these two daughter cells are quantitatively and qualitatively equal or similar. And they are two equal halves of their mother cell. That means the quantity and the chromosome number, etc. Like these characters. That is, among the two daughter cells, they are equal. And they are two equal halves of their mother cells, and their chromosome number is exactly similar to the mother cells. So the division is known as equational division. Mitosis generally known as equational division. And here the next division, which is meiosis, that is reduction division. Why the term reduction? Here the reduction is a reduction in the chromosome number. A reduction in the chromosome number. How much reduces the chromosome number? That is by half. Reduces the chromosome number by half, which means the parent chromosome number is 18. Then the daughter chromosome number is 9. Here the 18 is the diploid chromosome number and the 9 is haploid chromosome number, which means in the process of meiosis, the cell diploid nature reduces the haploid nature, and the product of meiosis, how many cells are formed during the meiosis? That is 4 daughter cells. The 4 daughter cells, that is the product of meiosis. So the direct differences among the mitosis and meiosis, the end product is 2 daughter cells in the case of mitosis, and the end product is 4 daughter cells in the meiosis. Then these two division, equational division, generally takes place in somatic cells. Hence it is known as somatic cell division. Somatic cells, which means vegetative cells. Our body cells, it is somatic cells. But in meiosis, that means the chromosome number reduces. Reducing the chromosome number, which takes place only in the case of reproductive cells or germ cells. So here this one is somatic cell division, and the meiotic division, and that type of division that takes place only in germ cells or sex cells, which means the formation of gamete through meiosis, the formation of our body cells through our size increases is due to the mitotic division. Next, offsprings or next generation that takes place, or that formation of next generation, or the cells required for the next generation, that is the germ cells or sex cells or gamete, that is due to the meiotic division. This is the main difference between mitosis and meiosis. And these two processes are complex process. But among these, meiosis is more complex than mitosis, but mitosis and meiosis, obviously it is a complex process. That means it consists more than one steps, or it is a step-by-step process. In the case of mitosis or meiosis, there are two types of division. First one is karyokinesis, and the next one is cytokinesis. That means whether it is mitosis or meiosis, it is completed due to karyokinesis and cytokinesis. Among these, the first process is karyokinesis, the next process is cytokinesis. Karyokinesis involves generally 4 steps: prophase, metaphase, anaphase, and telophase. So in the case of mitosis, the first step is prophase, then metaphase, anaphase, telophase, and cytokinesis. And the cell which is enter into the karyokinesis, that is definitely which is from the cell cycle stage. That means any cell enter into the mitosis or meiosis, first they follow or they perform cell cycle activities such as, or stages such as G1, S, and G2. So these G1, S, G2, these are sub-stages of a stage, a main stage which is known as interphase. Interphase. So a cell cycle which consists 2 stage, interphase and M phase. We already talked about the mitosis and meiosis, that is belongs to the M phase. So interphase converted or divided into 3 stages, G1, S, and G2. But some cells enter into an inactive stage from the G1 stage, which is known as G0. They are permanently or temporarily devoid the divisional capacity or metabolic activities. They are actively involved in metabolism, high rate of metabolic activity, but they are enter into a resting stage which is known as G0 or Kcet stage. So G1, S, G2 and M. This one is the stages of cell cycle. These 3 stages which is known as interphase, and the next stage is M phase. So whether it is mitosis or meiosis, the cell enter into 3 stages. Meiosis or mitosis, the cell enter into 2 stages: karyokinesis and cytokinesis. In the case of meiosis, there are 2 types of meiotic, or it involves 2 division: meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. Meiosis 1 involves prophase 1, metaphase 1, anaphase 1, then telophase 1. After there is a cytokinesis 1. So the cell which undergo meiosis 1, obviously it is a diploid cell. And after cytokinesis 1, they produce 2 haploid cells. That means the reduction division which takes place in the meiosis 1, and hence this meiosis 1 is also known as actual reduction division. Meiosis is the reduction division, but the actual reduction division takes place in the meiosis 1. And these 2 haploid daughter cells enter into the meiosis 2. Here also prophase, which is known as prophase 2, metaphase 2, anaphase 2 and telophase 2. And also there is a cytokinesis 2. So these two daughter cells enter into the cytokinesis. So they produce two daughter cells each. Hence the total daughter cells of meiosis is 4 and they are in haploid nature. So the division is known as reduction division. So this one is all about meiosis. But in the case of mitosis, only prophase, metaphase, anaphase, telophase and cytokinesis. Hence, which one is more complex, mitosis or meiosis? Basically, meiosis is the most complex process than mitosis. Among these meiosis 1 and meiosis 2, the longest stage of meiosis is prophase 1. Why? Why prophase 1 is the longest stages of meiosis 1 or meiosis? Because meiosis 1 or prophase 1 which contain many substages, or prophase 1 is divided into many other stages such as leptoteme, zygoteme, pachyteme, diploteme and diakinesis. The terms, the stages related to meiosis 1 or especially in prophase 1, they are leptoteme, zygoteme, pachyteme, diploteme and diakinesis. Leptonema, zygонема, pachyonema, etc. Then what is the characteristics of leptoteme? The characteristics of leptoteme is almost similar to the prophase or the starting of the prophase. Lepto, that means lepto, smallest, thin, hair-like structure. Chromosomes appeared as thin or narrow, hair-like structure. But the stages zygote, that is the fusion between two cells, that results in the formation of zygote. Here, zygotene, which means, or the term or the stage zygotene, here there is a fusion or pairing of non-sister chromatid of homologous chromosome. There are many terms: non-sister chromatid, homologous chromosome, then pairing or synapsis. Synapsis is the characteristics of the zygote. So, homologous chromosome, there are chromosomes, they are homologous. Homologous, homo means same. Logos, locus or location. The two chromosomes which have same gene location, which is known as homologous chromosome. Among these two chromosomes, one is derived from the father and one is derived from the mother. So there are two chromosomes. In a diploid cells, in our body, there are 46 chromosomes or 23 pairs. Is it right? Among these, 23 chromosomes coming from father and 23 chromosomes coming from mother. After the fusion, there are 46 chromosomes or 23 pair. So, the first chromosome of father and first chromosome of mother, they are similar because of their gene location. So, these two chromosomes having the same gene location, hence it is known as homologous chromosome. So, homologous chromosome is a pair of chromosomes. So, in a diploid cell, in our diploid cells, there are 23 pair. Each pair is a homologous chromosome. So, they are homologous chromosome. Then what is a non-sister chromatid? So, a chromosome, structurally, a chromosome which consists two chromatid, they are adjoining in a centromere. So, centromere, here this one is the centromere, and the yellow colored cylindrical part which is chromatids. Here, these chromatids are sister chromatid. These two chromatids of a chromosome is sister chromatids. Here there is a homologous chromosome. So, first one is the chromosome derived from father. This one is the chromosome derived from the mother. So, there are two chromosomes and these are homologous chromosomes. The first two chromatids of this chromosome, this one is sister chromatids. Similarly, these two are also sister chromatids. A homologous chromosome which contain sister chromatids. And these two chromatids, the chromatid of this first chromosome and the chromatid of the second chromosome, these are known as non-sister chromatid. So there are two terms, sister chromatid and non-sister chromatid. Obviously, sister chromatid, one chromosome, they have two chromatids and these chromatids are sister chromatid. Non-sister chromatid, the term related only in the case of a homologous chromosome and they have same gene location, but these are the chromatids of two chromosomes. They are known as non-sister chromatid. Here, the fusion or pairing that takes place in between non-sister chromatid of homologous chromosome by the help of a protein which is known as synaptonymal complex. Synaptonymal complex, it is a cementing material or it is a protein that is formed during the zygote which helps in the synapsis or pairing. So after the pairing, these two chromosomes, they are adjoined with or paired with each other and generally it is known as bivalent or synapsed chromosome. So what are the character of zygote? Zygote characteristics with the synapsis or pairing. The next stage is the packaging. Packaging is the most important stages in the meiosis. It is because of the function of meiosis that is takes place during the packaging stage. Okay, moving to the next question. The water potential of pure water. The question is coming from transport in plants. Water potential. Water potential or psi W. Psi W is equal to psi S plus psi P. Here the psi S is solute potential, psi P is pressure potential, which means water potential is the sum total of solute potential and pressure potential. Here the water potential is the ability of water. Solute potential is the ability of the solute to change the water potential. Pressure potential is the ability of pressure to change water potential. So, the sum of the solute potential and pressure potential is water potential. Water potential of pure water under normal temperature and normal atmospheric pressure is 0, which means if the solute potential is always negative, so if the solute potential is -5, if the water potential of pure water, that is 0, then the pressure potential is plus 5. So the sum of solute potential and pressure potential is 0. Then water potential of pure water under normal atmospheric pressure and temperature is 0, but when the pressure is changed, that means if the pressure is changed to 10 plus 5 to 10, then the water potential is also changed. That means it is a sum total of solute potential and pressure potential. The main or major terms related to the water potential or water characters, that is water potential. So water potential of pure water is not always 0, is under normal atmospheric pressure and normal pressure and potential, that is 0. So it is not 1, it's not more than 1, it's 0, it's not less than 0. Generally solute potential is negative, so pressure potential is also positive. Then pressure potential, one of the most important or most important pressure potential is target pressure. That means it is the pressure to help the cell to turgid. So that is always a positive. So generally pressure potential is positive, solute potential is negative, so water potential is 0. But under the conditions or under the change of this pressure and solute potential, water potential is also changed. So it may negative or positive. So water potential is 0. Then moving to the next question. Which match the pigment given in list 1 with their color in chromatogram given in list 2? There are certain chlorophyll pigment, not only chlorophyll pigment but also some other pigments and their color. So color, the color of leaf, color of flower, etc., it is due to some pigment. That pigment, some certain pigment which give a definite color. It is because of the presence of this coloring substance or protein. The first pigment, that is chlorophyll B, then carotenoids, chlorophyll A, xanthophylls. Based on the color, there are some pigments that is known as chlorophylls. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and also chlorophyll E. Then carotenoids. Generally, these carotenoids are categorized into two groups, carotene and xanthophyll. Carotene and xanthophylls. Dacus carota, that is the carrot. Carrot color, orange color, yellow orange. Carotene, that is yellow orange. Chlorophyll A, that is the blue-green color. Chlorophyll A is the major pigment of blue-green algae and also green algae, chlorophyceae. So they give a blue-green. Then chlorophyll B, which gives yellow green. Xanthophyll, which gives yellow. Chlorophyll B, which gives yellow green. Carotenoid, which gives yellow orange. Chlorophyll A gives blue green, and the xanthophyll gives yellow. So the right answer is option B. 1T, 2P, 3S, and 4R. So the answer is B. So moving to the next question. Hydra reproduces sexually by producing What type of structure? In this chapter, Living World, the first chapter, Living World. In this chapter, we study the characteristics of living organisms such as growth, reproduction, metabolism, cellular organization, and also consciousness. The major characteristics of living organisms are growth, reproduction, metabolism, cellular organization, and consciousness. Among these, growth and reproduction is not considered as the important character, and metabolism, cellular organization, consciousness. These are the main characteristics of a living organism. Then growth and reproduction, they are considered as the synonym in the case of unicellular organisms such as bacteria. That means in the case of bacteria, what is growth? What is reproduction? Growth is the number, increasing in the number of cells. Reproduction is the production of new offspring, that is also increasing in the number of cells. There are different type of reproduction. Mainly they are categorized into two: asexual reproduction and sexual reproduction. There are two type of reproduction. They are asexual and sexual. Asexual reproduction, which means there is no involvement of two parents. There is an involvement of only one parent, and also there is the complex process such as gametogenesis, gametic fusion, and also zygote formation or embryo formation, etc. But in sexual reproduction, there is an involvement of two parents, and they produce two gamete, and there is another process which is known as fertilization, and after fertilization, the zygote developed into embryo or embryogenesis, etc. Among these, the asexual reproductions, some of the examples: asexual reproduction, that is spore formation. The most important example is fungus. In fungus, their main reproduction method is spore formation. They produce different type of spores. Not only fungus, many primitive organisms, they produce different type of spores. And these spores, they are categorized into different types of spores: planospores, conidiospores, etc. Conidia, there is another type of asexual reproduction method of fungus. The next type of asexual reproduction is bud formation. Budding. They produce bud. The organism which produce bud, they are hydra. And also the only unicellular fungus, that is yeast. The main mode of asexual reproduction is budding in the case of yeast and hydra. Then sponges, and another type of organism, that is sponges, their main reproduction method is gemule formation. So the gemule formation, bud formation, spore formation, binary fission. Like bacteria or yeast like organism, they reproduce by binary fission or cell division. So many primitive or smallest organism, they reproduce asexually by different means such as spore formation, budding, then gemule formation, etc. Here the question is hydra, their main asexual reproduction. Hydra reproduce asexually by producing bud. Okay, so the answer, not spores, conidia, gemule, they are wrong answer. Then the budding or bud, that is the asexual reproductive mode of hydra. So the answer is C. Okay. Then moving to the next question. It is coming from, this question is from second year chapter, that is a sexual reproduction in flowering plants. Here the question is, the role of filiform apparatus in synergid is 2. Filiform apparatus in synergid. So what is synergid? What is a filiform apparatus? And this question from sexual reproduction, it is related to a structure which is known as embryo sac. Embryo sac. A typical angiosperm embryo sac is 7-celled and 8-nucleated. An angiosperm embryo sac which is characterized with 7-celled and 8-nucleated, and these 7 cells are categorized into 3 groups which is known as egg apparatus. An egg apparatus which consists one egg cell, then 2 synergid or synergids. So egg apparatus which consists one egg cells and 2 synergid. So there are 3 cells. Among the 7-celled, 3 cells are egg apparatus. And the second group is antipodals. The chalacal cells of the chalacal region of the embryo sac, which consist 3 cells. They are known as antipodals. So here 3 cells, here 3 cells, so they are 6 cells. The next, a central cell, the large cell of the embryo sac, which is central cells. Central cells is one-celled in nature, but it consists 2 nucleus. One cell with 2 haploid nucleus. So there are 1 cell. So 3 plus 3 plus 1, there are 7 cells. Each cells which consist 1 nucleus, so 7 celled, but the central cells which consist 2 nucleus. So an extra nucleus is present in central cells, so it is 8 celled. So why it is 7 celled and 8 nucleated? Because of the central cells which contain 2 haploid nucleus, which is known as polar nucleus. They are known as polar nucleus because they are coming from each pole. One coming from chalacal region, one is coming from micropylar region. So this one is the structural characteristics of an angiospermic embryo sac. Then these structures which helps in the fusion of gamete, okay, or these are for the fusion of gamete, embryo form, then zygotic development, etc. So this one, thus this embryo sac which consists egg cell. This egg cell fused with the male gamete. Okay. The fusion of male gamete and the egg cells resulting in the formation of diploid zygote. And this embryo sac which helps in the zygotic formation, and that zygote develop into embryo formation. So egg cells and male gamete, when the pollination takes place and the pollen grain reaches the embryo sac and it releases the male gamete. When this male gamete releases, these egg cells fused with the male gamete. So the function of egg cells, that is the formation of zygote. Except these egg cells, there are 2 synergids, 3 antipodals, then 1 central cells. The main characteristics of angiosperm is double fertilization. Double fertilization. Double fertilization, which means there is 2 fusion or 2 fertilization. One fertilization that is known as syngamy, that is the fusion of male gamete with the female gamete, egg cells with the male gamete. Then second one is the triple fusion. Triple fusion, there are 3 fusion, 3 cells, fusion of 3 gamete or 3 cells or 3 nucleus. There is a fusion of male gamete with 2 polar nucleus. This fusion of 2 polar nucleus with a male nucleus leads to the formation of primary endosperm nucleus, hence it is triploid in nature. Triploid, ploid is changed to triploid. Haploid condition, diploid condition, and there is another condition which is known as triploid condition, which means if the haploid condition is 4, then the diploid condition is 8 and the triploid condition is 12. So, primary endosperm nucleus is triploid in nature. It is because of these are the product of triple fusion. So, syngamy leads to the uh, diploid condition. Triple fusion leads to the triploid condition. So, there are two fusion which takes place in the angiosperm. So, they are known as double fertilization. So, double fertilization involved in the cells which are involved in the double fertilization, egg cells, male gamete, then two polar nucleus. So the remaining cells are synergids and antipodals. What are the role of these synergids and antipodals? According to NCERT, these synergids and antipodals, after the double fertilization, these cells undergo degeneration or degenerate. However, there are many functions. They are performed by antipodals and synergids. The main function of synergid. The main function of synergid which helps in the entry of pollen tube into the embryo sac or up to the egg cells. Synergid which guide the entry of pollen tube towards the embryo sac or egg cells. So the role of filiform apparatus in synergid to protect the egg apparatus, not endosperm formation. It's wrong. Prevention of gamete entry. No. Here the role of filiform apparatus which helps in the or which guide the entry of pollen tube to the embryo sac for the fusion of or for the syngamy and triple fusion. So here the answer is option C. So moving to the next question, that is also coming from sexual reproduction in flowering plants. Transfer of pollen grain from the anther to the stigma of another flower of the same plant. Here the transfer of pollen grain from the anther of one flower to the stigma of the same flower or another flower, that process is known as pollination. Pollination, the first process of sexual reproduction in angiosperm or gymnosperm, higher plants. There are different ways of pollination, zoophilic or hydrophilic, anemophilic, endomophilic, etc. But this process takes place in one plant or different plant or one flower or different flower. Based on that, they are categorized into different. They are directly categorized into homogamy or heterogamy. Homogamy, which means it takes place in one plant. Heterogamy, which takes place in different plant. That means there is a chance to pollinate within a plant or within a flower or in between two different plants. So within a plant or homogamy takes place by means of two ways: autogamy or geitonogamy. Auto means same or self. Autogamy, if a plant having flowers and they are in bisexual condition, which means which contain stamens and also carpel, there is a chance to pollinate itself. That means the transfer of pollen grain from anther to the stigma of the same flower. Anther to the stigma of same flower. That phenomenon is known as autogamy. Then geitonogamy is if the plant which contain bisexual or unisexual flower. There is a chance, bisexual or unisexual. There is a chance to transfer the pollen grain from anther to the stigma of different flower of the same plant. The transfer of pollen grain from the anther to the stigma of the different flower of the same plant. There is one plant, but pollination takes place between the different flower of that or the same plant. That process is known as geitonogamy. And this phenomenon takes place in different plants also. That means if the plant is a that bear unisexual flower, or the plant is dioecious in nature. One plant is male plant, another plant is female plant. There is pollination. The pollination takes place, or that should happen only from flower of the one plant to the flower of the another plant. That means that takes place in between two plant, not only two flower, two plants. So two flower also and also two plant. So one male flower transfers pollen grain to the female plant or female flower of the same species of the another plant. So this phenomenon is known as xenogamy. So autogamy, that takes place, pollination takes place in one flower. Geitonogamy takes place in two flower of same plant. Xenogamy takes place in two flower of the two plant or two different plant. Then there is another process is known as cleistogamy. Cleistogamy is a condition that autogamy process. Cleistogamy takes place only in cleistogamous flower. What is a cleistogamous? The flower do not open. Do not open. So, if the flower do not open, then the pollination takes place from that flower or itself. So, this is a type of autogamous condition, but the flower do not open. So the question is the transfer of pollen grain from the anther to the stigma of another flower of the same plant. Here, this one is the same plant, and the flower pollination takes place from one flower to the another flower. So that phenomenon is known as geitonogamy. Xenogamy, that takes place in two plant. Autogamy, one plant, one flower. Cleistogamy, one plant, one flower. Geitonogamy, one plant, two flower. So the answer is D. Then the next question. The natural reservoir of phosphorus is. This question is coming from ecology part, directly from ecosystem, the chapter ecosystem. In this chapter, there are two types of nutrient cycling. Sediment cycle or gaseous cycle and gaseous cycle. Sedimentation or sediment cycle and a gaseous cycle. Carbon is a gaseous cycle. Phosphorus, sulfur, etc. It's a sedimentation cycle. Why this one is classified into gaseous and sediment, that is based on the main reservoir. In the case of carbon, the main reservoir is actually a sea or ocean, but the carbon that is easily available from the atmosphere. So this one is gaseous cycle. But in the case of phosphorus and sulfur, it is coming under sedimentation cycle. It is because the main reservoir is phosphorus rocks and the sulfur rocks of the earth. So the natural reservoir of phosphorus is rocks, not soil solution. Soil solution contain this element. It is due to the mineralization process. Then detritus, not then atmosphere. Atmosphere, that is the natural reservoir of carbon, that is the atmosphere. So the answer is rocks. Then the next question is the annual net primary productivity. Annual net primary productivity of the biosphere is approximately. It is also coming from the ecosystem chapter. What is productivity? There are many terms. What is primary productivity? What is secondary productivity? What is net primary productivity and also gross primary productivity? These are the different terms we need to know. First, what is productivity? Productivity in terms of biomass or energy, that is the actual production. But in terms of productivity, it is related to the time period and also the area. So productivity directly determine the how much production takes place in definite area in definite periods. Generally, the time period which is based on the 1 year production. So 1 year production takes place. And the area generally considered as 1 meter. 1 square meter. So 1 square meter area, within a square meter area, within an year, how much production takes place, that is known as productivity. In the case of energy, that is kilocalorie. In the case of biomass, gram production. Gram meter square. So how much production takes place? That is productivity. Then what is primary productivity and what is secondary productivity? Primary productivity, which means production or productivity based on the producers. That means plant produces how much energy or how much biomass. Productivity based on the producer level is known as primary productivity. Productivity based on the consumer level is known as secondary productivity. So primary and secondary productivity. Then what is net productivity? Then gross productivity. Gross production, which means the total production per unit time, per unit area. Net productivity, which means the total production minus the usage. In the case of organism, the respiratory loss, the term R. The letter R, which means respiratory loss. So net primary productivity, which means the total production minus respiratory loss at the producer level. Gross primary productivity, which means total production at the producer level in a definite area in a definite time period. So net primary productivity is equal to NPP is equal to GPP minus R. Gross primary productivity minus respiratory loss is the net primary productivity. According to NCERT, the annual net primary productivity of the biosphere, which is approximately 170 billion tons. That means producer level production in an year, which is a huge quantity, that is 170 billion tons. Not million, it is 170 billion, 100 crores. So 170 billion tons of annual net primary productivity that takes place in the biosphere. It's an approximate value. So the answer is A. Then the next question is, which of the following based upon the principles of antigen antibody interaction? The second year chapter biotechnology, there are some new methodology or new process, that is antigen antibody reactions, then PCR, ELISA, etc. Then which among the following is based on the antigen antibody? PCR, polymerase chain reaction. Polymerase chain reaction is a gene amplification method. It's not related to antigen antibody. ELISA test. ELISA test is used for the determination of the AIDS. That ELISA, that is based on the antigen antibody interaction. Recombinant rDNA technology and gel electrophoresis. rDNA technology, gel electrophoresis. Both are recombinant DNA technology. So, which among the following based on the principles of antigen antibody interaction? That is ELISA. Answer is option B. Answer is option B.